Okay, so I'm gonna do my protein synthesis walkthrough for the extra credit, or just the project, sorry. So starting off with a little bit of background information before we get into the process of transcription. Some things that are going to be referenced is kind of like, so the TAC is the start DNA codon, and the enhancer is a part of a region of DNA which is in front of the promoter. And when this is activated, it'll speed up the rate of transcription. And then on this slide here, we have a photo of the TATA box, which is where transcription factors will attach to the DNA. And this is how these factors will have an understanding of where they'll be able to bind to this DNA. So starting the process, here we have the TATA box, as you can see right here. And then this is DNA polymerase, which is, and these are transcription factors. And this is where the initiation complex begins, and DNA unwinds to create pre-mRNA by complementary base pairing like TA, AU, and CG. And then, yeah, so going on to the next one. Here in this on the left, this is the pre-mRNA, and this is all the transcription process. And RNA polymerase will then bind around the transcription factors. These are the transcription factors on the antisense strand. The purple is antisense strand. And then together they're called the initiation complex, as I kind of said previously in the other one. And then the initiation complex will move down the DNA strand, separating it and stuff like that. And so this is just kind of the representation of it on a larger scale. As you can see, the pre-mRNA, the sense strand. The sense strand is a strand that it's the coding strand. It's the untranscribed DNA strand. So here we have, this is just an overview. At the top is the beginning of transcription, which is where DNA from nucleic acid language to RNA nucleic acid language, and RNA is produced in this strand over here, as you can see. So now moving on into this area over the left here, we have pre-mRNA being transcribed on the antisense strand, as I mentioned previously. And when the pre-mRNA strand has been completed, it'll separate from the DNA strand and it'll reattach and rewind, and the pre-mRNA will move on to splicing. And splicing is like the SNRNPs, which is the small nuclear RNA, and their proteins, and the spliceosomes, which is just several SNRNPs. And they just cut and paste genes, kind of in a nutshell. And then moving on to down here, we're going to focus on, I just mentioned the spliceosomes and the SNRNP. So now we're looking at RNA splicing enzymes. And this is where we're coming. So we have the start codons and the stop codons. So you see these aren't compatible anymore. And then the exons and the introns on the pre-mRNA strand. And the SNRNP is attached in the front end of the introns. And then we're moving down here, which is where the SNRNP they'll be attracted to each other, kind of like magnets. And then when four SNRNPs come together, that makes a spliceosome, as you can see right here. And the spliceosome will cut the intron loop out of the pre-mRNA, and the loops will ultimately be broken down by enzymes and then back into RNA molecules. And the nucleotides will be recycled, used to make the new piece of pre-mRNA, which is what happens over here. And they detach from the introns or the exons, and the introns are sad and the extrons are happy. Excuse me. And the four SNRPs combine to make a spliceosome, as I said. Spliceosomes remove and cut the intron loops. Sorry. And the intron loops are broken down, and the RNA nucleotides are recycled, which is used for the next RNA molecule to be made. And then moving up here, this is the end of transcription. We have the 3' A tail, and then above it is the mature RNA that leaves the nucleus through a nuclear pore and goes to the cytoplasm and ribosome, which is where we'll start with translation. And so we have the 3' tail, and then we have the 5G cap. And the 5G cap is added to the 5' end of the mRNA strand, which helps protect the mRNA in the cytoplasm, but also leads the strand to a ribosome, which is where they'll begin the next process. And the 3' A tail is connected to the end because it's a long strand of nucleotides, and this also helps protect them. And the enzymes can only attach the 3' strand or the 3 strand. And the length of the poly A tail depends kind of how long the mRNA is needed to quote unquote survive in the cytoplasm. So this is now, it's not pre-mRNA anymore, it's mature. It's matured into mature RNA. So now we're out of the cytoplasm and we are in the ribosome. As you can see, the smaller subunit of the ribosome, and we have our EPA, our APE, the large subunit of the ribosome split into 2. These are the 3 docking stations or sites, because it's kind of like you can think of it as a UPS system in a way. But the A site is where the arrival happens and RNA arrives. And then in the P site, the protein or polypeptide, where proteins are assembled. And then in the E, the exit, where the tRNA minus its amino acid will leave the ribosome. So now really beginning into translation. Ignore that part of transcription, that's from the other part. We begin the process of initiation, which is where the mRNA arrives and it enters on the 5' end and it enters to the A site. And the tRNA molecule, which is carrying an amino acid, will bind to the first 3 molecules of the mRNA, and the tRNA anticodon will bond to this mRNA. As you can see, it's kind of like going through, almost like you can think of it as a conveyor belt. And then below, we're beginning elongation. And the amino acid complex will move from the A site to the P site, which can also be referred to as translocation. It's just the name for what's called when it's moving. And the new tRNA amino acid will bind to the next in the A site. And they move on to the next site, but the amino acid from the first TNR strand will let go of its tRNA and attach the amino acid in the A site. And the bond between these 2 amino acids is a peptide bond, not a polypeptide bond, a peptide bond, which is what's happening here. And then on elongation, we have the tRNA is moved into the P site, and it has the 2 amino acids that we talked before, the peptide bond. And this will happen continuously until the chain of amino acids is created. You can see it's going to go through and create a chain. And then below, that's kind of that process going, and it repeats until the amino acid is created. This is what I was referring to. It'll happen over and over again until a chain of amino acids is created, and the enzyme in the cytoplasm connects with the tRNA amino acid. This is that enzyme. It's connecting. You can see it go down. And then in termination, the stop codon in the mRNA is reached. The process will stop right here. And the polypeptide chain will detach from the ribosome and go through folding and then do what it needs to do for the organism to complete its function. As you can see, we have a peptide bond form, and this whole thing is that's a single peptide bond. This whole thing is a protein or a polypeptide bond. And so as an overview, building a polypeptide happens through initiation, elongation, and termination. And initiation brings together the mRNA and the ribosome subunits and initiator. Then elongation adds the amino acid based on codon sequence, and termination stops the codon. And then let's go back to the beginning and review. Here we have the splicing. And mRNA splicing is post-transcription processing where eukaryotic mRNA needs work after transcription. And primary transcription means pre-RNA to create mature mRNA transcript. And then to our ribosome, ribosomes facilitate coupling of tRNA anticodons, and it has two subunits, large and small, with the A site, the P site, and the E site. The A site holds the tRNA carrying amino acid to be added to the chain. The P site holds tRNA carrying growing polypeptide chain, and the E site, an empty tRNA, leaves ribosomes from the exit site. So that's kind of an overview of protein synthesis.